Chapter 3 Intestinal Microbes, the Unseen World. The two most hazardous things an astronaut takes into his capsule on extended flights are his brain and his intestinal flora. A man is only what his microbes make of him. It is generally accepted among physicians and researchers that during intestinal upset and chronic intestinal disease, the normal harmonious state of balance between intestinal microbes living in the gastrointestinal tract is lost. It is important, therefore, that we have some understanding of the inhabitants of the unseen world. Before birth, the human intestine is free of microbes. From the moment of birth, however, a massive invasion of the gastrointestinal tract takes place, and it soon becomes populated with various types of microbes depending on the type of milk ingested as well as other environmental factors. Some of the microbial growth depends from contact with the mother's skin. Some originates from the air. If the infant is breastfed, more than 99% of all microbes in the intestines are of one type. As other foods are introduced, the baby develops a wide variety of bacteria. Studies have revealed that eventually more than 400 bacterial species live together in the human colon. The stomach and most of the small intestine do not normally harbor more than a sparse population of microbial flora. However, the number of microbes normally increases at the lowest part of the intestine, the ileum, because of its close proximity to the microbial-rich colon. In the healthy intestinal tract, intestinal microbes appear to live in a state of balance, and an overabundance of one type seems to be inhibited by the activities of other types. This competition between microbes prevents any one type from overwhelming the body with its waste products or toxins. Another important protective factor which works to maintain the sparse bacterial population of the stomach and upper small intestine is the high acidity of the stomach's hydrochloric acid, in which microbes cannot usually survive. In addition, normal peristalsis, waves of involuntary muscular contractions, sweeps many microbes out of the intestines to be lost in the feces, thereby decreasing their numbers. However, bacterial overgrowth in the stomach and small intestine can and does occur for various reasons, among which are 1. Interference with the high acidity of the stomach through the continual use of antacids. 2. A decrease in the acidity of the stomach such as occurs in the aging process. 3. Malnutrition or a diet of poor quality and the resulting weakening of the body's immune system. 4. Antibiotic therapy, which can cause a wide range of microbial change. A microbe commonly residing in the intestine without harmful effect may undergo a wide range of changes as a result of antibiotic therapy. Once the normal equilibrium of the colon is disturbed for any reason, its microbes can migrate into the small intestine and stomach, hampering digestion, competing for nutrients, and overloading the intestinal tract with their waste products. Quite early in bacterial overgrowth of the small intestine, the normal absorption of vitamin B12 is disturbed. There is considerable evidence that vitamin B12 is poorly absorbed when microbes multiply within the small intestine, prevent uptake by the ileum. There has been a long history indicating that bacteria and yeast have been involved in intestinal disease. As far back as 1904, an examination of the stools of children who were suffering with what appeared to be celiac disease revealed abnormally large numbers of fermentative carbohydrate eaters and putrefactive protein eaters, bacteria which were undoubtedly contributing to the disease process. The physicians making this observation proposed that although the normal intestine controlled the growth of bacteria in celiac disease cases, some intestinal abnormality prevented the normal regulatory control. Early researchers working on ulcerative colitis believed this disorder to be caused by bacteria. From 1906 to 1924, numerous researchers isolated certain types of bacteria, injected either the bacteria or the bacterial toxins into laboratory animals, and claimed that the injections produced ulcerative colitis in the animals. In 1932, one Dr. B.B. Crohn spoke about a new intestinal disorder which he called regional ileitis, now known as Crohn's disease. Some physicians attending his lecture stated that this new disease entity might be due to microorganisms. From the 1920s until the present, the role of microbes and the products they produce continues to be investigated in an effort to find the cause of the various forms of inflammatory bowel disease. Often there has been very convincing evidence that particular bacteria could initiate a certain type of intestinal disease, but eventually the work has been dismissed because of insufficient proof. Some of the difficulties which these investigators experienced in trying to pinpoint the culprit microbes were undoubtedly due to the ever-changing conditions of the microbial world of the intestine, to variably in the strains of intestinal microbes, or to the lack of precise laboratory techniques of identification. During these early years of investigation, Dr. Ilya Mechnikov proposed that bacteria in the intestine were producing toxins which were then absorbed into the bloodstream. These toxins, Mechnikov stated, were the cause of many human affliction, and he named the process by which harmful microbes in the intestine cause disease autointoxication. Unlike investigations that successfully attempt to define the precise microorganisms involved in the various types of intestinal disorders, Mechnikov approached the problem quite differently. He maintained, as many others have done, that the intestinal environment can be kept in a healthy state. Harmful microbes will no longer be a threat. He advocated the widespread use of acidified fermented milk, similar to yogurt, and proposed that the beneficial bacteria used in producing the fermented milk and still remaining therein would enter the intestinal tract and prevent other bacteria in the intestine from forming harmful toxins. While Michnikov's proposal has not been universally adopted, his ideas are acknowledged by outstanding gastroenterologists and researchers. In 1964, Dr. Donaldson stated in a lengthy article about the role of bacteria in the intestinal disease, in certain respects, the concept of autointoxication offered by Michnikov must now receive serious consideration. Investigators continued to be fascinated by Michnikov's proposal and to study the potential benefits of acidified milk. Modern researchers are asking, do the bacteria used to ferment the milk actually take up residence in the intestine? And if so, for how long? Which of the yogurt-type bacteria used to acidify milk will counteract toxins produced by other intestinal microbes? Is the bacteria used to acidify the milk, or the acidified fermented milk itself, the benefit factor? In the 1980s, an increasing number of reports have been published stating the intestinal bacterial toxins appear to be injuring intestinal cells, and as a result causing a variety of diarrheal symptoms. Some of the bacteria producing these toxins have not, in the past, been considered to be disease-causing types. Although there are still sufficient evidence to link a specific microbe to each of the chronic intestinal disorders, it is generally agreed that intestinal microbes are not innocent bystanders. A simple approach to minimizing the undesirable activities of intestinal microbes would seem to be through the use of antibiotics. This approach is often tried, but unfortunately in most chronic intestinal disorders it has limitations. We are forced then, with intestinal disorders which involve microbial populations which have been altered in numbers, in kind, or both. The normal contractions of the intestinal muscles are not able to remove them. They appear to be tenacious. Indeed, there is evidence that intestinal microbes will not cause disease unless they develop methods of adhering to the gut wall. Antibiotic therapy is of limited usefulness, while other drugs of the cortisone and sulfa families have side effects if continued too long. A sensible and harmless form of warfare on the aberrant population of intestinal microbes is to manipulate their energy, food supply, through diet. Most intestinal microbes require carbohydrates for energy, and the specific carbohydrate diet severely limits the availability of carbohydrates. By depriving them, intestinal microbes, of their energy source, their numbers gradually decrease along with the products they produce.